My name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure today to serve as moderator for this panel discussion titled, Ask the Experts, How Mycoplasma Contamination Affects Results and Reproducibility, sponsored by Biological Industries USA. I'd like to introduce our trio of panelists for today, starting with Tennille Ludwig. Dr. Tennille Ludwig currently serves as the director of the Ycell Stem Cell Bank, overseeing the banking, distribution, and core services operations at Ycell. Dr. Ludwig completed a PhD in embryology and developmental biology with a minor in bioethics from the University of Wisconsin-Madison in 2001. Her subsequent work in the laboratory of Dr. James Thompson focused primarily on the optimization of cell culture conditions and resulted in the development of the first defined feeder-independent culture system for human embryonic stem cells. Dr. Ludwig is a member of the Stem Cell and Regenerative Medicine Center at the University of Wisconsin, serves on the steering committee for the International Stem Cell Banking Forum and the International Stem Cell Initiative. Hello, Dr. Tennille. Thank you for joining us today.
Good afternoon.
Next is Seth Tapkin, the Director of Operations at Ycell. In this role, Seth manages organization-wide efforts that support the mission and goals of Ycell. Seth also leads the Characterization Laboratory at Ycell, which specializes in pluripotent cell cytogenetics. Seth holds a Master of Science in Biotechnology from University of Wisconsin-Madison and a BA Biology from Malacaster College of St. Paul. Seth also holds ASCP certification as a clinical specialist in cytogenetics. Prior to his tenure at Ycell, Seth worked at a diagnostic lab with Genzyme Genetics, gaining experience in clinical cytogenetics. Seth has previously worked in a project management for Epic Systems, a medical record software company, and in international development with the United States Peace Corps. Seth enjoys working closely with investigators and companies to understand their research needs and assist in designing a characterization plan utilizing Ycell's expertise to meet their unique need. Seth, thank you for joining us today.
Thanks.
Finally, with us today is Dr. Jessica Lawler. Dr. Lawler is a member of the Interdisciplinary Brands editorial team at Wiley and serves as developmental editor for 3 of the current protocols titles, Protein Science, Nucleic Acid Chemistry, and Toxicology, as well as the journal Wired System Biology and Medicine. She focuses on making research methodology more reproducible and accessible to scientists of all experience levels. Jessica received her doctorate in virology from Harvard University and has a strong research background in a wide range of life science subjects, including molecular biology, biochemistry, and genetics. Hello, Jessica. Thank you for being with us today.
Hi, Jeremy. Great to be here.
So with that, let us begin with a series of discussion points, starting with Dr. Ludwig.
Thank you so much, and we'd like to thank Current Protocols at Wiley and Biological Industries USA for the opportunity to participate in this collaborative conversation today about mycoplasma, the effect that it can have on your research, reproducibility, and your publications. To give you a roadmap into our conversation today, first we're going to talk a little bit about mycoplasma itself. What is it? How does it affect your work? We're going to talk about some tips for avoiding contamination within your laboratory. We'll talk a little bit about how to test for mycoplasma, and then we'll be turning it over to Jessica at Wiley, who can talk about problems in the laboratory and how those problems create concerns in publications, rigor, and reproducibility. We've already been introduced, but just as a reminder, I'm Dr. Tennille Ludwig. I direct the Stem Cell Bank at Ycell, and I'm joined today from Ycell by Seth Tapkin, who's our Director of Operations. For those of you who are unfamiliar with Ycell, we are a small nonprofit research institute located in Madison, Wisconsin, in the north central United States. We were originally founded along with the derivation of the first cell lines by Dr. Thompson in Madison, Wisconsin. So Ycell has essentially been around as long as human pluripotent stem cells have been around. We're most known for our banking and distribution of human pluripotent stem cell lines. We have more than 1,500 stem cell lines that are available for academic and government investigators all across the world. We pride ourselves on having high-quality, well-characterized lines. with a number of control models, disease models, reporter cell lines across a variety of platforms. We also have GMP clinically compatible cell lines and FDA drug master files in place to accompany those lines for groups that are interested in going into the clinic, and are very happy to say that those cell lines are currently in use in clinical trials. In addition to our cell banking services, we also offer a full range of characterization services, including a number of genetic screening tests like karyotype, fluorescence in situ hybridization or FISH, microarray or SNP array tests. We also do authentication testing or identity testing through STR analysis, and mycoplasma testing, which we'll be talking about in depth today. We pride ourselves on a rapid turnaround for karyotype, 7 to 10 business days, and we work to address, to provide these tests to help address rigor and reproducibility in publication work. If you need any more information on any of our banking efforts or characterization services, you can reach us at www.wycell.org. So on to today's topic, mycoplasma. What is mycoplasma and why should you care about it? Mycoplasma is a very, very small bacterium. It's 0.15 to 0.3 micromolars in size, so exceptionally small and can have a very variable shape. It is the smallest known prokaryote that's out there, and because it is so small, it is not easily visualized in culture. In fact, for my money, you can't see it. I've had some people tell me that they can visualize it in culture, but unless you have extraordinarily eagle eyes, you will not see this in culture. Because of that, it's a very easily missed infection. It is an incredibly resilient organism. It doesn't want to die. It will continue to perpetuate in your culture. It's completely completely resistant to some of the things that would normally damage a cell, including pressure, temperature, changes in osmolarity, dehydration. Its lack of a cell wall and the ability to not have a standard shape makes it not susceptible to the traditional antibiotics that would normally kill a bacterial contamination. And it's present both inside and outside the cell, and the fact that it's taken up into the cell is what creates a very dramatic effect on your cell cultures. So the dangers of mycoplasma and why should you be so concerned about it? The biggest reason is that it can affect the experimental results of your work, and it damages cells in culture. It will promote host cell death through pro-apoptotic genes. Essentially, it induces your cells to kill themselves. So you'll see, the one thing you may see is an increased amount of cell debris within your culture. It will compete with the host cells for nutrients because it consumes those nutrients itself and then basically starves the culture. It also can contribute undesirable metabolites and cytokines into your culture, and between this reduction in nutrients and addition of other metabolites, it changes your culture composition so that now you don't have a firm command or even knowledge of what your culture condition is anymore. And in doing so, it creates a situation where you may not be able to repeat or reproduce your results because you can't reproduce or repeat the exact culture conditions. It contributes to extremely high variability within your cultures, and it ultimately results in unreliable data. It can also induce genetic abnormalities and malignancies within your culture, and because we know that genetically aberrant cell lines do not perform in the same way that genetically normal cell lines do, it can also affect your experimental results in that capacity. It is a potential human pathogen, and so when we start talking about GMP and work that's destined for the clinic, this becomes a significant concern. And it can spread rapidly, and it can be very difficult to eliminate within your cultures. And it's really prevalent around the world. Now, the FDA has done studies to show that about 15% of all the laboratories within the U.S. are contaminated, but only 50% of the laboratories within the U.S. test. So half of the people that have mycoplasma don't know that they have it within their laboratories. 35% of the cell lines contained within cell banks are contaminated with mycoplasma. And these are just the reports coming out of the U.S. 35% of the laboratories worldwide are contaminated, and Japan has reported contamination results as high as 60%. So it's everywhere. And if this type of numbers doesn't scare you, it should. It really should. Where does this mycoplasma come from? Well, basically, you and your lab mates. We carry it with us into the lab every single day. 80% of people around the planet carry two types of mycoplasma, Mycoplasma salivarium or Mycoplasma orale. So it can come directly from you. One of the primary ways that it comes into the laboratory is if you're working with iPS cells, and those were created through human biopsy, it can come in on the primary material. It could have been infected at collection if the biopsy site was not prepared appropriately. It can come in through poor technique in the biosafety cabinet, and we'll talk more about that in just a bit. It can come in on cells from other laboratories that aren't doing testing on a regular basis. It can come in on cells that come from one of the cell banks that is distributing mycoplasma contaminated material. And it can be, even if you start out with every cell line in your laboratory being clean and you know that, you can infect them by talking or sneezing, laughing or singing, or all of the other sorts of fun things you do in your laboratory on a daily basis. So the symptoms of mycoplasma and how you know you're contaminated, well, the reality is you often don't. And that's where the concern comes in. A mycoplasma contamination is not obvious. You're not going to see the pH change that normally comes with a bacterial contamination that turns your culture media yellow. It won't happen. You're not going to see the turbidity that generally is associated with a fungal contamination in your laboratory. It doesn't do that. It's too small to see on its own. You won't see it swimming around like you do small bacteria. You just don't see it. You can see some evidence of it. It lives like a parasitic organism within your cell culture. It does overtake the cell culture, even though you don't see it, and to a really high degree. If in a contaminated culture, you can have 1,000 mycoplasma bacterium for every one cell of interest that you're culturing. What you may see, and it doesn't happen in all cultures, but you may see increased cell death. So you'll see the kind of sloughing of cells within your culture media that you might see after a double feeding on a weekend, but you would see it every day after a regular feeding. If you begin to see that level of cell death, that's one major sign to take a look. But other than that, the contamination is essentially invisible. So how do you avoid mycoplasma contamination within your lab? How do you avoid getting it to start with? Well, we strongly recommend that you quarantine and test all new cultures. And that doesn't matter where they're coming from, whether they're coming from a neighbor that you trust, a very well-respected cell lab, a cell bank, no matter where it's coming from, and no matter what testing they've told you they've done prior to you receiving it, quarantine it within your laboratory and test it prior to exposing other cultures to it. We really recommend a designated incubator, and if you can, a designated hood to work with it until it's tested clean of mycoplasma. Ideally, you would use a dedicated quarantine room and not move it anywhere into your general culture space until you had cleared it from mycoplasma. And this would apply to every new culture, every time, without exception. We recommend regular testing of active cultures. As we've already said, you can contaminate negative cultures within the laboratory, so you should establish a program of regular testing within your lab. We recommend at minimum monthly, preferably every 2 weeks, and as you do this, every culture within the laboratory should be tested. Proper hygiene. Wash your hands, change your lab coats, launder your coats on a regular basis. Use proper personal protection equipment, including gloves, face shields if it's necessary, and cleaning, wiping down your hoods on a regular basis, keeping the laboratory generally clean. Aseptic technique, we recommend working in obviously a biosafety cabinet, working as aseptically or as sterilely as possible. As you glove up and go into the hood for the first time, wiping down the hood with alcohol, wiping down your gloves with alcohol, and maintaining that aseptic technique. Work with only one culture in the hood at a time. This can prevent the spread of, potential spread of contamination from one culture to another. Once you're completed working with that particular culture, a quick wipe down of the hood with alcohol before the next culture is brought into the hood to process. Certify your hoods regularly to make sure that that their containment functions are working properly. This should be done on an annual basis. We still recommend that you limit talking within the hood. The hoods are certified for a standard airflow. Talking, depending on how you're doing it, and more importantly, coughing and sneezing, is not necessarily a standard airflow. Make sure your laboratory mates understand proper cough and sneeze protocol within the laboratory. It's winter. We get colds, and you can't keep people from working in the laboratory when they have a cold. But if you're going to cough or sneeze within the laboratory, we recommend that you push back away from the hood, turn away from it, cough into either the inside of your lab coat or into Kleenex or another barrier, and that you change your gloves before you re-enter the hood. We also recommend, again, that you launder lab coats on a regular basis. So I'm going to turn it over to Seth, and he can talk to you a little bit about determining testing for Mycoplasma.
Excellent. So the good news, after Tennille has told you all these scary things about Mycoplasma, is that we have both the ability to test for it and the ability to do so effectively and efficiently. So I'm going to tell you a little bit about how Ycell approaches the testing for Mycoplasma and a bit about our decision-making process around what assay to use and how we do it in our space. So when we are looking at the different options that you have for testing for Mycoplasma, they fall into a couple of easy buckets. The first being an enzymatic assay using luminescence. This would be, an example of this would be the MycoAlert assay from Lonza. This is a fast and inexpensive assay, and it is certainly better than nothing. It is, can certainly let you know when Mycoplasma or the enzymatic activity associated with Mycoplasma is present in your culture. The next way that you can assess your cultures for the presence of Mycoplasma is a PCR-based assay. Now, this is the method that Ycell currently uses in our fee-for-service characterization lab to assess cultures for Mycoplasma, the presence of Mycoplasma. That is both a sensitive and reliable way to detect Mycoplasma contamination. It is also being recognized by different regulatory bodies globally as an appropriate screen for Mycoplasma in the context of cell therapy. Now, at Ycell, we're not generally doing that testing for any kind of cell therapy. We're doing it for researchers and commercial labs who have a lot of cell culture. It is only to say that this is globally recognized as a good way to test for this particular infection. Last, there is the old gold standard of a 28-day culture assay for Mycoplasma. Obviously, this has its drawbacks. Any culture that's going to hang around for 28 days to be cleared of Mycoplasma is going to affect everyone's timelines, and of course has the ability of causing additional contaminations in the lab during that time. So I think for obvious reasons, this one is not as highly utilized as it once was. So Ycell, when we were looking at our testing options and trying to determine which kit we would use, we focused on a couple of things. We wanted a highly sensitive test that could be run at a reasonable cost with ease of use and great confidence in the results. What we ended up picking for our kit was the biological industry's Easy PCR Myco Detection Kit, We've been using it for a number of years at Ycell and have been happy with the results thus far. One of the things that we thought really was a good addition that this kit had that was meaningful for us was an internal control for PCR inhibition. This internal control provides a kind of a parallel PCR amplification that takes place right next to the amplification of the DNA from the mycoplasma and allows us to have great confidence in the fact that A, our amplification worked, B, a negative is indeed a negative for the mycoplasma contamination, and provides a layer of confidence with just general pipetting and the possibility for pipetting error in a lab where you're running a lot of samples and you're running those samples day after day, doing it very regularly. The kit that we use covers 90 species of mycoplasma. I believe it's about 5 species, including the 2 that Tennille mentioned, that account for 95% or so of all the mycoplasma contamination in cell cultures. So 90 species is perhaps overdoing it. As long as you've got those 5 species, you're probably in a good place with your testing protocols. But still to say, there are different species of this particular bacteria, and you do want to make sure that the kit you're using will pick up those species. And finally, we have a detection limit of 10 colony-forming units per mil. Now, Ycell has not validated the sensitivity of this particular assay down to 10 colony-forming units per mil. That we have not done. But what we have done is we've assessed the results that we've received doing the biological industry's PCR kit for myco against the luminescence enzymatic assays for mycoplasma contamination. And what we did find is that we were able to pick up mycoplasma contamination before, with the PCR assay before we were able to see it with the luminescence-based assay. That is not to say that the luminescence assay would not have eventually picked up that contamination. We expect that it would have. But what you gain in sensitivity is perhaps 1 testing cycle or 2 testing cycles earlier detection of a potential contamination in your lab, and that time can be very meaningful in the ability to curtail or stop a experiment or the culture of a given cell line in a lab space. Ycell has absolutely had the experience of thawing mycoplasma positive samples in our labs, and we take the decontamination process very seriously here. It really affects the work that can be done in a lab, and it affects technicians' time, stress level. There's a lot of things that go into a mycoplasma positive result in a given lab, and as Tennille spoke eloquently about the research implications, there are also just the staff implications and the resources that go into really cleaning something like that up. So we take it very seriously, and in our choice for a testing kit, we took into account the sensitivity and our ability to be really effective with the processes that we were putting in place. So rapid and effective. We found it, the PCR workflows that we use to detect mycoplasma contamination to be efficient in our hands, and we're able to generally provide results to people within a couple days of getting the sample. Understanding that we are working in a system where we will, run samples together as much as possible. So, moving on. What we have found in the testing that we've done in a fee-for-service context is that mycoplasma contamination does not discriminate. We work with a large number of different academic labs. We work with a large number of commercial labs. They are good labs. They produce good research. They do great work. But they all have had instances where there's been mycocontamination in their space, in their work. You know, when I looked back before doing this webinar at the number of samples that we've run and the number of positive results that we've gotten, you can see on this slide I'm putting up 3 to 5% of cultures showing mycoplasma contamination or mycoplasma infection. That's considerably lower than the percentages that Tennille provided earlier in her slide, where we might expect up to 15% of cultures coming from the U.S. to show this kind of contamination. What I would say is that the vast majority of samples that we are looking at are not coming in for mycoplasma testing. As Tennille mentioned, we do a lot of different genetic screening for cell cultures, generally around genetic stability, genetic fidelity. Is your cell line acquiring abnormalities as you keep it in culture over your protocols or gene editing experiments? So that's what we're doing a lot of the times. We do mycoplasma testing because we think it is important and we think it is a good baseline quality standard for labs to try to achieve. But all that said, a lot of groups are choosing not to use that resource or spend that money to do the mycoplasma testing here. Now, that's not to say they're not doing it themselves. I certainly hope they are. But in some of our genetic stability testing, we do metaphase preparations, which involve staining chromosomes to do karyotype analysis. What we do when we do that staining is we can actually visualize at 600 times magnification when we're doing our chromosome analysis. We can actually visualize mycoplasma on those slides, in those preparations, because the stain is staining the mycoplasma DNA as well. So we know when we're doing some of the other testing that we do that we are, that there are mycopositive cultures coming through our lab, and the testing on those is, not being done, at least in our lab. So what I think that means is that a lot of labs continue to not have a well-developed testing protocol, or if they do, that compliance to that protocol is, not 100%. So there are some antibiotics that exist that are effective against mycoplasma contamination or mycoplasma infection. Ycell has not tested these, and we really can't speak to the efficacy of these products. I think Tennille is fond of saying that bleach is our preferred treatment for mycoplasma positive cell cultures at Ycell, and that really has been our policy up to date. Now, I'm sure there are situations where a patient material might be the source of the mycoplasma, or some other really precious sample is showing mycoplasma contamination, where you might need to think about a regimen of antibiotics that could clear a culture of mycoplasma. To date, that hasn't been, we have not decided that that's been worth the effort here at Ycell, but one could imagine a scenario. Documentation for those antibiotics that I've seen suggests clearance protocols could take weeks, and of course cross-contamination would always be possible during this time. So needless to say, that's a lot of work, and that's a lot of additional testing that would need to be done to clear a cell line of mycoplasma testing, but it might be necessary in some cases. I'd like to wrap up our portion of the webinar with just some basic take-home messages here. Mycoplasma is worthy of significant concern in your lab. It does affect research results, and it will affect your reproducibility and rigor of the research that you conduct. We suggest strongly that you implement regular testing to identify and eliminate contamination. And we recommend strongly that you enforce practices to avoid contracting and spreading the contamination between your cultures, between your technicians, between your incubators, between your lab spaces. So training and follow-up is very important in controlling the spread of those potential contaminations. Thank you for your time. With that, I would like to pass it over to our colleague Jessica from Wiley.
All right. Thank you, Tennille and Seth, for the first half of the presentation. And so now we can move on to how mycoplasma contamination affects your results and reproducibility from the perspective of a publisher in terms of publishing reproducible and robust research for the community. And so for my half of the talk, I'll be covering first a slight introduction of myself, as they did before, and then we'll move into a history of mycoplasma contamination in the literature, then how mycoplasma contamination can affect your results and your reproducibility in general, and ways that it's been published that it does affect cell cultures, potential ramifications if for some reason someone did publish mycoplasma contaminated results, what the negative results could be, and then moving forward how we can eliminate this problem in future research. So as I said before, I'm Jessica Lawler, and I'm a developmental editor with Wiley in the Current Protocols group. I manage 3 current protocols journals in protein science, nucleic acid chemistry, and toxicology, and additionally a interdisciplinary review journal on systems biology and medicine. But slightly more important for this talk, I did my PhD work in virology and specifically at Harvard Medical School in a herpes virus lab. It's been known since the 70s that mycoplasma has a significant effect on herpes virus growth and the cytopathic effects that it has on cell culture. So the first thing that our boss always said when something went wrong for troubleshooting was, have you tested for mycoplasma recently? And the result was that most of the time that wasn't the problem, but it was a good thing that we were testing on a regular basis anyway. So jumping into a little bit of the history of mycoplasma contamination in the literature, we've actually known that mycoplasma contamination has existed in cell cultures since 1956. And you can see the science, a little bit of the science paper here, but it's actually a 1-page science paper, which I found was interesting, how publishing has changed over the years. And you can see the micrograph of the colonies on agar that they were able to find in their culture. And even more importantly, not only did they find this contamination in one of their cultures, but 4 of the 6 cultures that they actually tested from their lab were contaminated. So it was known pretty early on that this was going to be a major problem in the literature and in cell culture labs in general. And moving forward, there's been over 60 years of literature showing that mycoplasma affects our research and our data. Now, there's a lot of reasons why this why 60+ years of work hasn't resulted in a decrease in contamination, and as you've seen, the rates of contamination are still high. And some of the main reasons for that result from the difficulty they had in detecting mycoplasma, especially back in the 50s onward. There's also, at the time when they discovered it, there was no antibiotic susceptibility, so it was very hard to clear any cultures besides through bleach. And it can pass easily through cell culture from human contamination, and mycoplasma can actually exist within the liquid phase of a liquid nitrogen tank. So if by any chance you have a change in pressure and one of your tubes pops open that's contaminated in a liquid nitrogen tank, it could potentially get to all of the other tubes that you're cryo-storing in that tank as well, which is a major concern. And then next, not all labs are created equal. There are a lot of good policies out there for testing for mycoplasma, but it's not firmly used in every single lab, which has made it hard across all labs to get a certified policy and tools that people can use to limit mycoplasma contamination and effects on their data. So I did a short search just to see how many papers had been published on mycoplasma contamination in the Wiley Online Library, and you can see that I found over 8,600 different publications relating to this subject, suggesting that it's still a major issue in the literature. And actually, when I looked through PubMed, I realized that the number of citations mentioning mycoplasma contamination has been steadily increasing since its discovery in 1956. So it's still a big issue today and something that needs to be addressed by the community at large. Now, you might be wondering, that's all well and good, it's a big issue, but how does this affect my data? And I could go through the 8,600 plus articles describing how mycoplasma contamination affects your specific subject area, but I'll cover a few of the major ones and let you go out and do some research on your own. So the first thing you need to know is that mycoplasma infection can alter microarray gene expression profiles. Both some genes are upregulated and downregulated, and it's pretty widespread across the board. Additionally, if you're trying to grow any 3D cultures, such as 3D kidney cysts, it can affect your ability to form these different model systems. Next, even the 1,000 Genomes project was shown to have some mycoplasma contamination present, with up to 7% of the scans that they read through the sequencer actually belonging to mycoplasma contamination instead of human genome. Moving on, it can affect the growth of viruses, like I said before, and the cytopathic effects that it incurs on the cell culture it's infecting. Can affect interferon induction and susceptibility to cell-based killing for immunology. And actually, one of the more interesting things that I found while looking through the literature was that they, on purpose, contaminated some cell bioreactors to test how different protein expression, large-scale experiments have been affected by contamination of mycoplasma. So this is a problem not only in small labs, but in industry and larger labs that are trying to scale up development for therapeutics. Just a few more things I found is that it can affect development and maturations of different cell types, and can actually decrease the ability to diagnose different disorders if that diagnosis requires cell culture. And then, as I said before, cytopathic effect can affect infections of different kinds, including HIV-1, and it can induce oxidative stress and actually attenuate the DNA repair activity of cells. So really, mycoplasma contamination affects every aspect of cell biology, and it's really difficult to differentiate which data should be used and which should not when mycoplasma contamination comes into the picture. So it really should be a point of more talk and concern within the community at large. And while we've now seen that published data can be affected by mycoplasma contamination, what are some of the potential ramifications of putting this irreproducible data out there for the public? And most of the time this is, I wouldn't call it a negligible problem, but most of the time what happens is a lot of sleepless nights for trainees attempting to reproduce these experiments that aren't able to. However, there can be many different and worse ramifications of such a thing, such as retraction and addendums to published literature. I was looking through and was actually able to find at least a couple different cases where literature has been retracted due to mycoplasma contamination or potential mycoplasma contamination, rendering these studies not reproducible by the at-large scientific community. And actually, in another very interesting paper, they had published a brand new gene editing mechanism that that people were having trouble reproducing, and so the secondary authors that were trying to reproduce the data published a letter to the editor saying that they weren't able to do it, and the actual original authors suggested that maybe the reason they weren't able to reproduce was that there was some sensitivity to mycoplasma or bacteria in the culture. As it turned out, this was not the case, as many different labs that had been readily testing were not able to reproduce the results, but it was interesting that this was raised as one of the concerns through the eventual retraction of this paper. So the best way to deal with the effects of data through mycoplasma contamination are usually through addendums and retractions, but this is a risky system that doesn't always result in retraction or edits to the published literature. A lot of the time, retractions require that all of the authors sign off and are okay with the retraction before it goes through. So there are a lot of factors that play into this. And sometimes not all of the data present in the paper is not reproducible. Additionally, if we're spending time and effort and money using these results, then attempting to reproduce this data that's been affected by mycoplasma contamination wastes valuable resources and time that people could be using to better and forward research. And then there's also a negative stigma associated with retracting data. So there's a lot of people that are unwilling to take this step forward if they're worried it'll affect career trajectories. And again, it's hard to publish negative data in our current climate, suggesting that if people do have trouble reproducing data, that the attempts to reproduce are generally not published, whereas the original study stays in the literature. So there's a lot of concerns associated with publishing data that isn't reproducible and making sure that mycoplasma contamination doesn't make it into initial publications, because it turns out to be very difficult to further remove it from the public eye. So moving forward, the most important thing is, how do we fix this problem that seems to be impacting research on a global scale? And I've suggested a three-pronged approach to this, coming in through all of the different parties associated with academic research nowadays. The first being funders. This is an addendum to publishing grants sent out by the NIH in 2017, and you can see that they've specifically mentioned that authors need to be able to verify the cell lines that they use, specifically including mycoplasma testing for all of their cell lines. So funders are clearly seeing that this is a problem that they need to start addressing, and hopefully in the future we'll have more and more funders adding this as necessary to receive funding and be able to do the research in the correct way. The next is improving journal policies, and this is also something that has started, is in the process of getting moving for mycoplasma contamination. As you can see, there have been a number of different journals recently that have either required or encouraged that their authors verify their cell lines and test for mycoplasma before publication. And this number keeps growing yearly. But as of right now, not every journal requires this, and there's still a bit more work necessary to get everyone on the same page regarding this serious concern. And then finally, the biggest and best way to leave your impact on mycoplasma contamination and reducing it in the lab is by improving your own lab policies. We can talk all we want about reducing the data, the irreproducible data, and impacts on research, but prevention is really the best policy to keep mycoplasma from coming into your lab in the first place. And so we really recommend testing, testing, and testing again. Your cells need to be tested on a regular basis, and there need to be policies in place to make sure that this testing is robust and that the entire lab is taking part. Additionally, as said before, make sure to quarantine cell lines that come in from outside the lab. You never know what has touched those cell lines, and it's always important to confirm that everything you're using from outside your own lab is really what you think it is. Then again, check your controls, the classic scientific concern. Make sure that everything you're doing is controllable, and again, use good aseptic technique. And avoid talking, laughing, or sneezing in the cell culture room while you're working. So to summarize my half of the talk, we've seen that mycoplasma contamination has been a concern in cell cultures since 1956, and there are over 60 years of content describing the ways that contamination affects cell function and cell biology in general. And ignoring mycoplasma testing and contamination can affect the results of your research, the reproducibility of your results, and could ultimately lead to retraction if this is not addressed. Then finally, funders, publishers, and labs are working to decrease mycoplasma contamination, but it's an ongoing process that needs more attention and conversation throughout the scientific community. I want to thank you for listening, and we'll have a Q&A session coming up, but if you have any specific questions, feel free to contact me through email. All right, I'll bring it back to Jeremy for questions and answers.
All right, thank you, Jessica. And thanks to Tennille and Seth for their really good presentation. So some of the questions that we're getting, I'd like to actually start with Tennille. So one of the things that people are wondering is, so really, how do you know if your cell source is reliable and trustworthy? And kind of related to that is, when you do receive a new cell line, like how soon should you look to test that for mycoplasma contamination?
Very good question. So in terms of, how do you know who to trust? Trust no one. I trust no one. I even recommend that people don't trust me when we send material out. You should, even though I'm sending you a cell line with a certificate of analysis that says that it is mycoplasma clean, you should still quarantine it and mycoplasma test it in your laboratory. As far as how soon after you've thawed it should you test it, you're generally testing with a cell supernatant. We recommend, at Ycell, we want it tested within 3 days of thaw, but that all depends on how the material was cultured prior to you receiving it. If it was cultured with antibiotics, there is a withdrawal time. The antibiotics will interfere with your ability to detect the mycoplasma, and it depends on what the antibiotic is, how long the withdrawal time is. It can be between 5 and 14 days. So look into that, find out what your withdrawal time is, culture your cells in isolation until that point, and then test it.
Thank you. So somewhat related to that, in terms of, so Seth, this might be a question for you. So in terms of how good is mycoplasma at surviving things like cryopreservation? And then subsequently, when you thaw your cultures, what effects are you looking for?
Right. So I think Jessica mentioned it in her slides, but the ability of mycoplasma to live through being stored in liquid nitrogen, cryopreserved, and that it can actually contaminate other cultures if the tube ruptures and the mycoplasma is in the liquid nitrogen in liquid phase, it can move to the other tubes. So obviously it survives that environment well. During thaw, and how you would actually know if it was mycoplasma positive or contaminated with mycoplasma post-thaw, you wouldn't. And I think that's that's really a reason to kind of focus on this particular contaminant as a rather insidious, difficult thing to detect, and that's why we're suggesting these regular testing protocols as really being the only way to confidently say that you're culturing cells without mycoplasma contamination.
Thank you. So another question that has arisen is that, so many cell lines, especially stem cell lines like that, they can be greatly affected by their environment and things that are happening in the culture. So are there known effects on gene expression with mycoplasma contamination?
I'm not sure specifically about specific cell lines, but in general, there is a overall dramatic effect on gene expression because there are effects on the nutrient level available to the cells and the actual presence of the infection. The cells change their immune signaling, their metabolic rates, and their metabolic pathways, and basically every aspect of cell biology can change after mycoplasma contaminates a culture. So I'm not sure about any specific cell, and you can probably go look up a paper that'll tell you the results for that, but it really does affect every cell type and in every media.
Yeah, I mean, I think that this really drives home the concept that you really need to do this kind of testing. So when the thing that you do not want to have happen happens, and you actually do discover a mycoplasma contamination at Ycell, what do you actually do to clean that up? Not just in the cultures, but I mean, I imagine the equipment must be, you have to really kind of take care of the equipment as well.
Yes, and we have received cell lines from, very reputable laboratories that weren't aware they had it, that are mycoplasma positive. We're very fortunate in that we have a quarantine room so that it's isolated away from our banking sections. Because we are a cell bank, because we are distributing cell lines globally, we really have to institute a take-no-prisoners approach to a mycoplasma infection, which is why bleach is my primary treatment method. Not only do we bleach the cell culture, but out of an abundance of caution, we will bleach everything else that shared an incubator with it, and we will bleach everything else that touched the same hood that it touched from the time of the last mycoplasma test forward, or from the time of the thaw forward if it's a newly thawed material. After that, we will completely decontaminate the incubator, which includes taking it apart as much as possible, wiping everything down with alcohol, autoclaving the unit, putting it back together. We will completely decontaminate the hood, microscope, any surfaces within the laboratory, and then we institute sentinel cultures, which is essentially a known mycoplasma-free culture that we will culture in that hood and incubator for 2 weeks with mycoplasma testing, and nothing is released for general use again until it's tested mycoplasma clear. And we took down the whole quarantine laboratory at one point because cells had been cultured in the same hood as cells with another incubator, so we had to institute another quarantine lab. But the problem is you can't see it, and so that's the big question. How do you clean up something you can't see? We take a very firm hand at it, and we really recommend that if you see the same kind of contamination in your laboratory, that you institute something similar. And that's why it can take such a long time to get a laboratory back up and functioning after you've identified a mycoplasma contamination.
Great, thank you. So along those lines, I mean, you've cleaned everything up. People in the lab are now just using the best sterile techniques that they possibly can, taking every preventive measure. Do you still then need to test on a regular basis?
Absolutely. We test every 2 weeks, and we test every culture within every incubator to assure that we are maintaining a mycoplasma-free status within the laboratory, and we think that's absolutely critical in being able to provide confidence not only to ourselves but to those who are receiving materials from us. But again, don't trust me. Test it yourself when you receive it.
So turning a little bit now to kind of, just the mycoplasma detection itself. So Seth, you discussed some of this. Can you just kind of reiterate some of the important factors when choosing a detection method but then also what led you to discover that the most effective kit for what the most effective kit to use was for this mycoplasma detected at Ycell?
Sure. So the factors that we considered in our assessment was sensitivity, ease of use, reliability, as in can our technicians and our lab staff run the protocols, without error? Is it relatively straightforward protocol? And then the positive and sort of negative control aspects of the assays. Those are the things that we really focused on. With sensitivity, we had a suspicion that we were interested in a PCR-based assay as opposed to the enzymatic luminescent type assay. That said, we've used MycoAlert in our labs at times in the past with good results, but we thought in the context certainly of a fee-for-service testing lab as well as our own banking efforts, we thought the increased sensitivity that a PCR assay provided was a good idea, was advisable. And so sensitivity there was really important. Again, I'll mention the internal control that the biological industries kit provides, because I think that was really a differentiator with regard to some of the other kits out there. I don't know that we looked at another kit that provided an internal control for the actual PCR amplification, and that's just a really nice control to have. As I said, it provides us additional confidence in the fact that there is nothing in the media or in the experiment that has caused the PCR inhibition or the amplification to fail for some reason. It provides us that level of confidence, and it also provides the technicians who run the test regularly with a little added confidence that there was not a simple pipetting error during the test. So we're running these amplifications, and then we're running gels. So it's not quantitative. It's a is it there or is it not kind of a test. And if you are looking at the absence of evidence to be your negative, that's a little scary for me as someone who's doing this test for other labs who will rely on it. So it's important, we think, to have that internal control, and we think that's a meaningful differentiator for the BI kit.
Great. So, Jessica, this might be a difficult question to answer, but maybe it'll help drive home some of the points. Are there really any good ways to determine when your data has been contaminated by Mycoplasma?
I'm sorry to say this, but there really isn't. The best way is if you've discovered Mycoplasma contamination in your lab, and if you're testing regularly, you can at least go back in time and say, all right, the contamination happened between this date and this date, therefore any data within that time is suspect. But when you're looking in the literature, unless the data's been retracted or has been amended in some way, unfortunately, there's really not a good way to figure out which data has been affected. So I would recommend, if it happens in your own lab, make sure that you're testing regularly so that you know when this contamination occurred and which of your data is trustworthy. And then when you're looking in the literature, be skeptical of everybody. Try to reproduce any crucial results that you're using moving forward in your lab. And again, be wary of everything from any other lab that might touch your lab as well.
So, Jessica, you did mention that a lot of journals, a lot of publishers are looking for this Mycoplasma testing. And, this is kind of just a general comment question for everyone. Should publishers really institute very strict regulations to show that your cultures are not contaminated with Mycoplasma?
I can start off. Personally, it's Jessica here, sorry. Personally, I think that it should be a more widely used system, and it should be readily known which labs test for Mycoplasma and have tested over the course of the data collected for their papers and which haven't. I think it'll take a bit of work from policymakers and editors and even labs themselves to actually implement said policies, but it really is more important than the current conversations in research would lead you to believe. So while I think this is starting to become an issue, I think it's going to even become more of a point of talk in the future, and hopefully we can set policies in place that'll make this not a worry for scientists in future years.
And so our kind of wrapping up last question for the day is really just for everybody, and it would be great if everybody just kind of give a quick comment about it, is, for you, and in your opinion of the work that you do, why is contamination still such a large issue worldwide, and what can be really done to address this in the future?
I'd be happy to start. I think it's an issue worldwide because it's historically an issue, and we're dealing with, the distribution of cell lines that have probably long had these contaminations or these infections with Mycoplasma, and the fact that some cell banks are still distributing samples that have Mycoplasma infection that's a tough thing to get around until everybody changes sort of their approach to this testing and culling those samples that do show it. Beyond that, I think it's a simple resource issue for a lot of labs out there. What are they have limited funds and limited time, and what are they going to spend their time on? Again, the difficult thing about Mycoplasma is its insidious nature. It is both impossible to see through regular phase microscopy, and then its effects are, as we've described here, they can be rather subtle in some cases, and so it is difficult to really know that you need to do this work. It's not as easily seen as it could be, or as we'd like it to be. Let's say that.
And I agree with Seth. I think part of the issue is that you can't see it, and so it's sometimes easy to forget that it could potentially be there. You don't want to find out that you have a Mycoplasma contamination after you've published your results and nobody can repeat them. So getting into a normal routine is something that every quality lab should be doing, and you should be testing on a regular basis. It's just we haven't had a lot of conversation around Mycoplasma in the industry in a long time. So kudos to the publishers who are now looking at this with a larger microscope, looking at, and to the NIH for putting standards around rigor and reproducibility. So we believe that there are certain tests you need to do routinely, karyotype, identity, and Mycoplasma, and that those should be done prior to the initiation of any experiment and at the conclusion of any experiment to assure yourself as a researcher of the validity of your results.
And from my opinion, there's a lot that can be done, mostly from the policymakers, but additionally, just every researcher needs to be aware of their impact on the community, and they really need to own their data and be aware that this is a problem that can affect everyone. So we really all need to work together to test everything that's coming through. Let people know if you find something that's Mycoplasma contaminated. Maybe they weren't aware that their lab had this problem and you can fix it. We test every CRISPR-edited cell line out there, so Mycoplasma should be about the same.
All right, well, we've reached the end of our panel discussion for today on how Mycoplasma contamination affects results of reproducibility. I'd like to thank Dr. Tennille Ludwig and Seth Tapkin of the Ycell Cell Bank and Dr. Jessica Lawler of Current Protocols for their time and insight. This presentation was sponsored by Biological Industries USA in partnership with Wiley's Current Protocols. This webinar will be available for on-demand viewing on the Current Protocols website at www.currentprotocols.com in the webinar section. Thank you for attending, and thank you to our speakers for a great presentation.